Good morning, class. Today, we will be diving into the fascinating world of medical nanotechnology. In this lecture, we will explore the intricate design and engineering of targeted drug delivery systems, the use of stimuli-responsive nanosystems for on-demand drug release, the effectiveness of nanoscale carriers for intracellular drug delivery, and the critical importance of nanotoxicity assessment and safety considerations. Additionally, we will discuss how nanomaterials can be engineered to control drug release kinetics, the development of controlled drug release systems for ocular drug delivery, the use of nanoemulsions and nanocapsules for oral drug delivery, and the exciting advancements in nanosystems for targeted therapy in cancer. We will also explore the bottom-up and top-down fabrication techniques utilized in creating various nanosystems, the emergence of theranostic nanosystems for simultaneous drug delivery and imaging, the potential of nanobiohybrids as controlled drug release platforms, and the revolutionary impact of nanosystems on controlled release of gene therapies. Finally, we will conclude our lecture by summarizing the key points discussed and emphasizing the significant role that nanosystems play in the future of medicine. Nanotechnology has revolutionized drug delivery by offering precise targeting and enhanced efficacy. Designing targeted drug delivery systems requires a comprehensive understanding of the physiological barriers and pharmacokinetics involved. The process typically involves encapsulation of drugs within nanocarriers, such as liposomes, polymeric nanoparticles, or dendrimers. These carriers can be modified with ligands to bind specific receptors on target cells, ensuring selective drug release. Careful engineering of the carrier's physicochemical properties can control drug release rates, biodistribution, and cellular uptake. Additionally, nanosystems can protect drugs from degradation, increase their solubility, and enable sustained release over prolonged periods. By optimizing the design and engineering of targeted drug delivery systems, we can enhance therapeutic outcomes while minimizing off-target effects and toxicity. Stimuli-responsive nanosystems act as smart carriers that release drugs in response to specific triggers. These triggers may include changes in temperature, pH, redox potential, or the presence of enzymes in the target site. By incorporating stimuli-responsive components into the nanocarrier structure, drug release can be precisely controlled. For example, pH-sensitive polymers can be used to selectively release drugs in acidic environments, such as solid tumors or inflamed tissues. Temperature-responsive nanoparticles can be engineered to release loaded drugs when exposed to mild hyperthermia. The ability to trigger drug release on demand reduces the side effects associated with systemic drug administration while ensuring maximum therapeutic efficacy at the target site. To tackle diseases at the cellular level, efficient intracellular drug delivery is essential. Nanoscale carriers can successfully transport drugs across cellular barriers and facilitate their release inside target cells. For instance, nanoparticles can exploit endocytic pathways to enter cells and deliver their cargo. Once inside, the carriers promote drug release in response to specific intracellular conditions. These carriers may take advantage of the acidic pH in endosomes to trigger drug release or utilize enzymatic activity within lysosomes. The ability to deliver therapeutics directly to the intracellular compartments increases drug concentrations at the desired site of action, ensuring maximum efficacy. While nanotechnology offers tremendous opportunities, it is crucial to address the potential toxicological effects of nanomaterials. Nanotoxicity assessment involves evaluating the safety profile of nanosystems and understanding how they interact with biological systems. Factors such as nanoparticle size, shape, surface charge, and composition can influence their toxicity. Assessing the biocompatibility of nanomaterials requires comprehensive in vitro and in vivo studies to determine their impact on various cellular functions, such as viability, proliferation, and gene expression. Safety considerations include minimizing the release of toxic byproducts during degradation, understanding potential long-term effects, and ensuring proper disposal of nanosystems. By thoroughly evaluating nanotoxicity and implementing safety protocols, we can confidently harness the potential of nanotechnology for biomedical applications. Controlling drug release kinetics is a crucial aspect of designing drug delivery systems. Nanomaterials can be engineered to manipulate drug release rates, ranging from sustained release to pulsatile or stimuli-responsive release. In drug-loaded nanoparticles, the drug can be embedded within a polymer matrix or adsorbed onto the surface of the nanoparticle. The release kinetics can be tailored by adjusting the polymer composition, molecular weight, and polymer-drug interactions. For instance, hydrophilic polymers tend to promote sustained release, while hydrophobic polymers can facilitate burst release. By exploiting the unique properties of nanomaterials, we can finely tune drug release kinetics to optimize therapeutic outcomes. Traditional ocular drug delivery methods face numerous challenges, including inefficient drug penetration and rapid clearance. Nanosystems offer a promising solution by providing controlled drug release directly to the targeted ocular tissues. Nanoparticles, liposomes, or hydrogels can be used to encapsulate drugs and ensure sustained release over extended periods. These nanosystems can be engineered to improve drug bioavailability, increase residence time in the eye, and enable targeted delivery to specific ocular tissues. By overcoming the limitations of conventional drug delivery methods, nanosystems open up possibilities for effective treatments of ocular diseases. Oral drug delivery is the most convenient and patient-friendly route. However, it often faces challenges such as low drug solubility, poor stability, and limited absorption. Nanoemulsions and nanocapsules have emerged as promising platforms for enhancing oral drug delivery. Nanoemulsions consist of droplets dispersed in water, stabilized by surfactants or polymers. These droplets provide a large surface area for drug dissolution and facilitate transport across the gastrointestinal tract. Alternatively, nanocapsules are solid or liquid nanocarriers encapsulating drugs and surrounded by a shell. This shell can protect the drug, enhance its solubility, and control its release. Incorporating nanosystems into oral drug delivery can improve drug absorption, enhance bioavailability, and offer targeted release to specific regions of the gastrointestinal tract. Cancer therapy often faces challenges due to the lack of specificity and high toxicity of traditional treatment methods. Nanosystems have emerged as powerful tools for targeted therapy in cancer. By functionalizing nanocarriers with ligands recognizing specific cancer cell biomarkers, targeted drug delivery can be achieved. These nanosystems can selectively accumulate in tumor tissues, allowing higher drug concentrations at the target site while reducing systemic toxicity. Additionally, nanosystems can enhance the delivery of various therapeutic agents, such as chemotherapeutic drugs, gene-based therapies, or immunotherapeutic agents. Through the precise targeting and delivery of therapeutic payloads, nanosystems hold great promise in revolutionizing cancer treatment. The fabrication of nanosystems can be achieved through two main approaches: bottom-up and top-down fabrication techniques. Bottom-up fabrication involves the assembly of small molecular components or nanoparticles into desired structures through self-assembly processes. This approach allows for precise control over the size, shape, and composition of the resulting nanosystems. On the other hand, top-down fabrication involves the reduction of bulk materials into nanoscale structures using techniques such as milling, lithography, or laser ablation. This approach offers the advantage of scalability and compatibility with high-throughput production. Both bottom-up and top-down fabrication techniques play a crucial role in developing nanosystems with tailored properties for specific biomedical applications. Theranostics combines therapeutic and diagnostic functions within a single system, enabling personalized medicine and real-time monitoring of treatment efficacy. Nanosystems can be engineered as theranostic platforms by incorporating both therapeutic agents and imaging probes. For instance, nanoparticles can be loaded with anti-cancer drugs and conjugated with imaging agents such as fluorescent dyes, quantum dots, or radioactive tracers. This allows for simultaneous drug delivery and visualization of the biodistribution, pharmacokinetics, and therapeutic response. By integrating drug delivery and imaging capabilities, theranostic nanosystems provide a powerful tool for precision medicine and personalized treatments. Nanobiohybrids represent a novel class of nanosystems that combine synthetic nanomaterials with biological components to enhance their functionality. These hybrid systems offer the advantages of both synthetic nanomaterials and biological entities, resulting in synergistic effects. By incorporating biological elements, such as proteins, peptides, or cells, nanobiohybrids can achieve precise control over drug release and therapeutic activity. For example, nanocarriers can be coated with cell membranes to improve biocompatibility, evade immune detection, and achieve targeted drug delivery. The integration of biological components into nanosystems opens up new possibilities for controlled drug release and personalized medicine. Gene therapies hold immense potential in treating genetic disorders and various diseases at the genetic level. Nanosystems provide an ideal platform for the controlled release of gene therapies, ensuring efficient delivery and minimizing off-target effects. Nanoparticles or viral vectors can be engineered to encapsulate or deliver genes to target cells. These nanosystems protect the therapeutic genes from degradation and enhance their cellular uptake and intracellular delivery. By precisely controlling the release kinetics, tissue targeting, and gene expression, nanosystems enable the effective delivery of gene therapies and pave the way for the future of precision medicine. To summarize, medical nanotechnology offers tremendous potential for revolutionizing drug delivery and advancing personalized medicine. We explored the design and engineering of targeted drug delivery systems, stimuli-responsive nanosystems, and nanoscale carriers for intracellular drug delivery. Additionally, we discuss the assessment of nanotoxicity and safety considerations, the engineering of drug release kinetics using nanomaterials, and the implementation of controlled drug release systems for various applications, including ocular drug delivery and oral drug delivery. Moreover, we explored the exciting advancements in nanosystems for targeted therapy in cancer, as well as the fabrication techniques used, such as bottom-up and top-down approaches. We delved into the concept of theranostic nanosystems, nanobiohybrids, and nanosystems for controlled release of gene therapies. Through continued research and development in the field of medical nanotechnology, we can unlock new frontiers in drug delivery, diagnostics, and personalized medicine, ultimately improving patient outcomes and quality of life. Thank you for your attention, and I look forward to further exploring this fascinating field with you.